Hi everyone, my name is Varsha, and I was working with Isa Rodriguez on assessing CRISPR guide site viability and downstream transcriptomic effects in CFTR or cystic fibrosis editing. So, CRISPR is an incredibly popular and new scientific discovery within the last decade, and it can be easy for the general public to form preconceived notions of its effectiveness without actual science-based reasoning. So in an attempt to bridge this gap, my study essentially aims to show the database reality of this incredible tool. So, cystic fibrosis, or CF, is a genetic disorder that's caused by mutations in the CFTR gene. The CFTR protein normally functions as a chloride channel, which helps regulate the movement of salt and water in and out of cells. So, without it, mucus in the lungs and other organs becomes abnormally thick and sticky, which can lead to clogged airways, or chronic lung infections, digestive problems, and eventually progressive organ failure. So right now, patients manage cystic fibrosis through daily treatments like chest physiotherapy, inhaled antibiotics, and enzyme supplements and CFTR modulators. But there are major limitations to this. These drugs have to be taken for life. They're also extremely costly, and they don't work for every single CF mutation. And most importantly, they don't repair the underlying genetic effect. So this is essentially where CRISPR comes in. CRISPR, or Cas9, is a gene editing tool that works like molecular scissors. So it cuts DNA at precise sites that are chosen by a guide RNA. So for a single gene disorder like cystic fibrosis, CRISPR essentially seems like the perfect solution. Instead of managing symptoms, we can essentially directly repair the mutation at a DNA level. And a one-time edit could, in theory, restore healthy CFTR function permanently. So this vision has captured enormous public and scientific interest in the last couple of years. So the key questions of my study were, can we actually target CFTR with CRISPR? The gene is huge, it's spread across 27 exons, and mutations occur all over the place. So we need to know if there's enough safe and high quality CRISPR cut sites. And then second, what happens to the body when we remove or disrupt CFTR? So even if we technically succeed, would altering this gene create unintended downstream problems? So essentially, my project had two phases. The first phase was the guide site analysis, and for this, I used a web tool called ChopChop to scan the human CFTR gene. So for each exon, ChopChop essentially identifies all potential 20 nucleotide guide RNA sites and ranks them by quality. So guides are color coded by green, red, and amber. Green meaning high specificity, amber meaning moderate, and red is low. So for this study, I focused on the green or the best sites as viable candidates onto whether the CRISPR guide had essentially the highest probability of being successful. I also calculated an R9 score to see which exons were relatively more CRISPR friendly, which I'll explain further in the next slide. So finally, I compared these exons to the most common cystic fibrosis mutations to see if mutation hotspots actually did overlap with guide-rich regions. So the second phase was the transcriptomic analysis, and for this, I analyzed the RNA-seq data from a published experiment where CFTR was knocked out in mice. And this allowed me to compare gene expression in knockout versus normal cells. So for this, statistically significant genes were grouped into like functional pathways, and one out of the 15 pathways that were disrupted, I then examined in detail. So the first key finding is that CFTR is extremely hard to target with CRISPR. Out of all 27 exons, only 20 had zero high-quality green sites. And only one exon had more than one viable green site. And out of all of them, only over 96% of the exon had at most one green site. So the figure right here shows that each exon is a black box, and the dots above it essentially like represent potential guides. So almost all the dots you can see are red or amber, and the green sites are almost non-existent. I also used a score, which is shown in the second graph, to represent the ratio between the number of target sequences and the size of a given exon to essentially understand how the CRISPR guide distributes the target sequences, which I named the R9 score. So when I compared the guide-rich exons with the most common CF mutations, there was almost no overlap. So for example, the most common CF mutations, which is F508 del in exon 11, lies in a region with almost no viable guide sites. So technically, the CFTR gene is what we might call a CRISPR desert. It resists precise editing because it simply doesn't offer enough safe cut sites, which means that a universal CRISPR cure for cystic fibrosis is actually not feasible with our current tools. So if you try and edit only the exons with the green sites, you've achieved maybe a partial correction at best, which wouldn't cure the disease for sure. And if you resort to red or amber sites, you also risk dangerous off-target mutations elsewhere in the genome, which was the second part of my study. So here, the second key finding is that knocking out CFTR in this gene causes widespread transcriptomic disruption. So in the RNA-seq dataset, we found hundreds of genes significantly altered after CFTR loss. So these were grouped into 15 key pathways, and they included disrupting functions like lipid metabolism, detoxification, oxidative stress management, and immune signaling. So this shows that CFTR is not just a chloride channel for sure. It's a regulatory hub that's integrated into multiple biological processes. So removing it actually destabilizes a lot of critical cellular processes. So one example that I was looking at was the LPS IL-1 mediated inhibition of RXR. And RXR, or retinoid X receptor, is a nuclear receptor that essentially regulates genes involved in cholesterol balance, fatty acid metabolism, inflammation, and detoxification. So when CFTR was knocked out, downregulated genes were the ones responsible for cholesterol transport and fatty acid oxidation, which suggests a higher risk of things like metabolic disorders, cardiovascular disease, and oxidative stress. And at the same time, several inflammatory diseases were actually upregulated, which reflects a lot of heightened immune activation, beneficial in short bursts, but it can actually be harmful if it's chronically sustained. So, what does all of this actually mean for patients? Even if CRISPR could fix cystic fibrosis, the act of editing could actually trigger side effects resembling a CFTR knockout. So patients could exchange a lung disease for a chronic system inflammation, increased cardiovascular risk, insulin resistance or diabetes, neurological complications that are tied to oxidative stress regulation. So in practice, this could actually mean a net decline in the quality of life, not an improvement like a lot of people think. So first, this raises a lot of ethical concerns. There's this danger of false hope because families may see CRISPR as a permanent cure when in reality the risks and limitations make it incredibly unsafe right now. And second, there's a lot of equity concerns. If only a handful of patients, based on their genes with CRISPR-friendly mutations, can benefit while others can't, this can actually deepen a lot of health disparities. Um, and third, there is the broader principle of just because we can cut the genome doesn't mean we should. So we have to remember that responsible science requires us to wait until safety and efficacy are both clearly demonstrated. So what are these safer paths moving forward? Some options right now are things like CFTR modulators, because while they're imperfect, these drugs have proven to have measurable benefits without actual genomic risk. mRNA therapy, which delivers healthy CFTR mRNA and can restore protein function without actually cutting the DNA. Pre-implantation genetic testing essentially prevents CF transmission without germline editing. And things like future CRISPR improvements, things that we can improve like base editors, prime editors, novel Cas enzymes that can one day maybe offer safer or more precise corrections of things like the cystic fibrosis mutation. So To conclude, technically, cystic fibrosis is nearly untargetable by CRISPR due to the lack of high fidelity sites. Biologically, knockout causes a lot of widespread dysfunction across a lot of key pathways, and ethically, the risks outweigh the benefits, and false hope needs to be avoided. So, the most responsible conclusion is this. For cystic fibrosis, the question is not just can we cut the gene, but should we cut? And based on current evidence, I think that the answer is no. Thank you for listening to my presentation, and I hope you enjoyed.